to immunity in endodontics. The role of immunity in endodontics. The immune system is very complex in humans. So, it's a complex system. These are two types, innate and acquired or adaptive type. In innate type of immunity, it consists of cells and molecular elements which acts as barriers to prevent dissemination of bacteria and its products into underlying connective tissue. The cells providing innate immunity are neutrophils, monocytes, eosinophils, basophils, dendritic cells and odontoblasts. Coming to the second type, that is acquired or adaptive, it invokes the release of specific receptor molecules by lymphocytes that which recognize and bind to foreign antigens. The cells mainly produce this acquired or adaptive immunity are T lymphocytes that which release the T cell antigen receptors and B lymphocytes that release B cell antigen receptors. Coming to the histopathology of periapical tissue response, the root canal of teeth contains or consists of numerous irritants, and mainly the infected root canals consist of a major number of microorganisms in it, which the root canals acts as a host for the microorganisms. So these infected root canals, which consist of numerous irritants in the form of microorganisms, penetrates into the periapical area, which forms or leads into the formation of periradicular lesions. Due to the unlimited source of undifferentiated cells and that which participates in inflammation and repair in the periapical area, and also because of the collateral supply, there will be seen an extensive destruction in the periapical tissue. So, the infection that which passes from the infected root canals and from the root canals into the periapical tissue causes much more destruction when compared to the pulp. Coming to the endodontic implication, Fish described the reaction of periradicular tissues to bacterial noxious products of tissue necrosis and antigenic agents from the root canal. Fish experimented the foci of infection in guinea pigs by drilling openings in the jaw bones of the pigs and packing them with the wool fibers saturated with broth culture of microorganisms. In 1939, Fish described the zones of infection or the reaction of the body to infection. He concluded that removal of nidus of infection can resolute or leads to resolution of the infection. The first zone is called zone of infection, where the infection is present in the center of the lesion. The microorganism present only in that center area, and it consists of dead cells, destructive components and end products, protein decomposition products, etc. And the cells which are predominantly seen in this zone are polymorpho-neutrophils, that is leukocytes. Coming to the second one, that is zone of contamination, where the principal different cells are round cells, lymphocytes, the macrophages seen in less number. There will be a dilution of toxic elements. The cellular distortion around the central zone is appreciated in this second zone, and there will be an autolysis of bone cells forming empty lacunae, and this histopathological appearance represents the stage of repair. Here in this zone there will be a start of repair. Third one is zone of irritation. The toxicity diminishes as distance increases from the central zone. The principal cells are macrophages and osteoclasts, and also we can see undifferentiated cells join to form osteoclast here, and the phagocytosis of collagen fibers by macrophages is seen predominantly in this zone. The leukocytes fills the gaps between the osteoclast for healing and repair. And the capillary proliferation is seen in this zone. The last zone is zone of stimulation. The toxicity is reduced, the fibroblast, osteoblast is seen, and this zone takes care of the periphery. The collagen fibers are laid down by fibroblasts that which act as wall of difference around the zone of irritation. That means this wall differentiates the zone of stimulation from irritation zone, the scaffolding on which osteoblasts lay new bone tissue. So here we can see the new tissue formation, regeneration. And Fish concluded that root canal is the seat of infection. So we can gain the knowledge from the fish study and can apply it for better understanding of reaction of periradicular tissues to a non-vital tooth. So the metabolic products and toxins released by the microorganisms diffuses into the periradicular tissue, means from the infected root canals. So if there is an increased number of microorganisms, we can see the abscess formation or start of abscess formation. And these metabolic byproducts and toxins that which released by microorganisms, they destroy the periradicular tissue, and also they are responsible for the release of proteolytic enzymes that which results in the pus formation. The third one, on the periphery of the lesion, there will be a stimulation of fibroblast that which build fibrous tissue, and osteoblast that will form the sclerotic bone. So once the root canal has devoid of microorganisms, means if it has the infected root canal has been treated, that which acts as a reservoir of bacteria, and if it is thoroughly cleaned, shaped and obturated, and that can lead to repair of periapical area and also periapical bone. So here the endodontic therapy includes non-surgical endodontic treatment and surgical endodontic treatment. Non-surgical endodontic treatment consists of access preparation, cleaning and shaping and obturation. The surgical endodontic treatment, under here the rationale of surgical endodontics is to remove the diseased tissue present in the canal and around the apex and retrofill the root canal space with biologically inert material so as to achieve a fluid type seal. It involves a retrograde preparation here. So that's how we conclude this rationale of endodontics. An occupation had to secure a theoretical grasp of the phenomena with which it is dealt, as the inflammatory process is the first and immediate response to any harmful stimulus. At this juncture, the clinician should intervene, and with thorough knowledge of the process, he should render the best possible treatment to the affected site. This completes our rationale of endodontics, and thank you for listening.